This is a cell culture room. It is a place for general routine of any molecular biology lab, and because of that, it gains great importance for biomedical research scientists. A laminar flow cabinet or hood. It is a main feature when you need to preserve sterility in your cell cultures. Once it is ready, the hood produces filtered airflow, which helps to avoid that any contaminant particle could settle on our cells. We need a wide range of reactives to perform different cell assays, and they should be ready for use and properly preserved. Some of them are kept at 4 Celsius degrees, like culture media, optimum, DMM, or RPMI, fetal calf serum aliquots, trypsin, mitomycin, ficol, or in some cases even phosphate buffer. Instead, some others need -20 Celsius degrees preservation, as non-decomplemented FCS, culture media stocks, glutamine, antibiotics, or a great amount of aliquots. Mammal cells are kept in incubators to maintain more or less 37 Celsius degrees. So if you want to change medium, first you should increase its temperature when taken from fridge. Everything okay? Then let's start work. But remember that anything entering the hood must be sterilized with alcohol, even your gloved hands. Cells are usually cultured in flasks like these ones. To access to them, it is required to remove the cap before, and from that moment on, you must watch what you do and perform a good practice protocol. So don't you pass your hands, arms, and tools over the cells. Pipetors are always useful when you work with cells, just because any assay needs a previous homogenization. Thus, aliquots become an equal among them. So you have finally decided to test whether or not this special drug of your project is truly effective against tumoral cells, isn't it? Well, if you want to compare effects prompted by different treatments, then each one of them should start with the same number of cells. Then you have to count cells. All of them! No, just kidding. You only have to count a little quantity using a Neubauer chamber, a glass cover, and 15 to 20 microliters of cells. After that, go to an optic microscope and look for the true Neubauer chamber. Hey, wait a minute! I already have it! No, indeed, that's just the glass support. You only can see the chamber focusing with a microscope. It's there, composed by 4 quadrants in the corners, and each quadrant including 16 small squares. Count cells in these quadrants and use this easy formula, and voila! Now you know how many cells you have in all your culture. However, microscopes can be used for many more things. Perhaps you need to see how your cells are going in your culture, before, meanwhile, or after the treatment. These are tumoral B lymphocytes, and they are okay. How do I know it? Look, they are perfectly grown. They shine, and mainly they do not aggregate themselves. They are alive and ready to treat them. Whoa! Do not forget centrifugation. If you need to change cell medium, then you have to do centrifugation. And it is important the rotor never gets imbalanced. So you must set tubes with the same weight, one in front of the other. Be careful with this step, because the centrifuge is a very sensitive and dangerous equipment. In the centrifugation, meantime, let's prepare some other reactives. Sometimes you have to prepare a dilution from a non-sterilized powder reactant. A filter like this one on a syringa can save the day. That's it. Steady, steady, because with this kind of things, there's no need of haste. Centrifugation is off, and all the cells are at the bottom of the tubes in those white pellets. Don't you see it? Just discard the medium of the tubes on this glass with bleach. It will kill any naughty cell who decide not to stay with their sisters at the bottom of the tube. One quick move and nothing of splashing, eh? Medium at 37 Celsius degree, and the calculation you did before. Well, now it's only a matter of concentration to avoid to spoil everything. So the same volume of cell must be seated in each well of these plates, and after that add a different treatment to each one. Rubbish goes to special trash bins, prepared against biological hazard. But remember, you are the responsible in charge while you use the facilities, and not every waste goes to the same bin. At last, the final step. Plates are taken to an incubator, which provides the right conditions to cells to grow. Let's hope the treatment will work out alright in a couple of days. And this is the lab routine we have to face every day. Tomorrow will be like this too.